Variant Visualizer can be accessed by clicking on the Genome Tools tab or button on the RGD homepage. Clicking on the link will take you to the strain selection screen, where you can choose the strains that you wish to compare. Once the strains have been selected, they will appear on the left screen. The strains begin in the order in which they were selected, however, you can drag them to change the order. The order they are in once you hit continue will be the order they are in once you reach the information screen. The variant data source is given as a lab symbol in parentheses following the strain symbol. This is indicated by an abbreviation for the institution such as RNAAS for the Royal Netherlands Academy of Arts and Sciences and MCW for the Medical College of Wisconsin. After hitting continue, you select the region either by chromosome and position, or by entering gene or marker symbols to delineate the region. Alternatively, you can enter a list of gene symbols, one per line or separated by commas or spaces. These need not be located in a single genomic region. Then you can select more parameters at the Option Select screen, which will further narrow your results by, for instance, predicted protein coding type, allelic call, or genomic position, although these are all optional. Within a category, the separator for multiple choices is OR, meaning that you could search for homozygous or possibly homozygous and get the results of each type of SNV, whereas between categories it is AND. So, if you choose both synonymous and premature stop codons, it would look for SNVs that meet both criteria, and you would get no results because an SNV that gave you a premature stop codon would by definition be non-synonymous. Only SNVs matching the options you have selected will be displayed on the next screen. When you reach the main screen, there are 3 links at the top left of the page. The first of these, the Home link, will return you to the strain selection screen. Clicking Region returns you to the region selection screen, including gene and position selection. This allows you to maintain the selected strains while reselecting region and options. In addition to these, the link Options returns you to the option selection screen, allowing you to maintain the selected strains and the selected region while reselecting the options. More towards the middle of the top bar of the screen are links named Variant Distribution and Annotate Region. Variant Distribution will display a chart that shows a total count of variants for each gene and/or intergenic region for each strain in the selected region. The gene or genes with variants that match your selected criteria will be displayed on the x-axis, while the strains will remain on the y-axis. This view is also the default for a region that has too many SNPs to fit in the normal screen, the cutoff being 2,000 SNVs, or for a list of genes that are not in the same genomic region. However, if you have searched for a single gene, then by default the individual variants for the entire gene will be shown regardless of the number of SNVs. Annotate Region links to the Gene Annotator tool, which is designed to display RGD annotations for all genes in the selected region. If you need assistance navigating the Gene Annotator, there is a tutorial video that can be accessed under the RGD Community Videos page. Just below these links, the chromosome, start, and stop position as defined for the current chart are shown and can be changed in the display. There is also a position count as well as a display of the region size. The first line of the display shows conservation scores. The closer the conservation value is to 1, the higher the probability that the base is conserved across species. This is conveyed visually as well, with the highest conservation scores being in the darkest red colors. Conservation scores are imported from FAST. Below that is the gene track for the plus strand. The genes are displayed as shaded boxes on the tracks, which are sized according to the number of SNVs. This particular region has 2 overlapping genes on the minus strand. If the box is too small to display the entire gene symbol, you can mouse over it in order to display the symbol. Selecting a gene will reset the region to that gene's start and stop position. It will also show the gene description. Gene descriptions are automatically derived from RGD's ontology annotation. The gene track for the minus strand works in the same way as the track for the plus strand. Below the gene tracks, the reference sequence track is viewable. The sequence that is referenced for this example is RGSC 3.4. Bases that fall in exons are highlighted with a dark background in this track. Below the reference sequence track, positions for each SNP are given in base pairs. In the strain variant tracks, a display with different bases indicate heterozygosity. 2 of the same base indicate homozygosity, and 1 base indicates that the variant was of unknown zygosity. The bases are color-coded by nucleotide, with adenine being dark blue, cytosine being light blue, guanine being orange, and thymine being red. The beige nucleotides represent spaces where there has not been a variant called for that position, either because the bases do not vary or because there was not enough data to declare said base to be variant. Heterozygotes are a combination of the colors of the 2 alternate bases. Selecting an individual base will display the variant details. These include basic information about the variant, including the sequencing read depth and the percentage of those reads that were read as the variant nucleotide. Genetic variants display the relevant RNA nucleotide accession number and the location of the variant in the gene. If the gene has multiple splice variants and therefore more than 1 nucleotide accession number, all of them will be shown. For variants in coding regions, the genomic sequence has been translated to show the amino acid sequence in which the variant amino acid is marked in red. For a quick guide to the variant visualizer, there is a basic help graphic that is displayed when you click on Help in the upper right-hand corner. Thank you for watching this presentation. More specific questions about this tool can be fielded to rgd.data@mcw.edu.